Yeah, you just switched it.
Looking good. Yes, look good. Yeah.
A quick land acknowledgement to Berkeley Public Library buildings are built on the territory of Fuchila, Chinese-speaking Ohlone people. We honor the 5,000-year history of communities of the West Berkeley Shell Mound and current Ohlone communities in the city of Berkeley and throughout Alameda County and the East Bay. The library recognizes that Berkeley residents have and continue to benefit from the use and occupation of unceded stolen land, and that the namesake of our city benefited from and advocated for settler colonialism and the forceful conversion of indigenous children to Christianity. Berkeley Public Library is committed to helping create a more just and anti-racist society and furthering the visibility and celebration of Ohlone authors and culture in our collections and programs. Welcome to Pop in the Science Bubble. This is a monthly seminar series that aims to share new research findings from grad students and postdocs at UC Berkeley with the general public. We have 2 speakers at each seminar who will talk about their current research or topic they find interesting. Organizers are grad students at UC Berkeley, Palin, Daniel, and Madison. Lectures happen on the 3rd Tuesday of the month at 5:30. These programs are also live streamed on our YouTube page, so you can view them later and share them with others.
Thank you again for showing up for our first Popping the Science Bubble of 2024, back after a little bit of a hiatus. So our first speaker tonight is Shubham Maurya. So Shubham started his graduate studies in India, the Indian Institute of Technology, Kanpur, by engineering viral vectors for efficient gene delivery. He moved to the University of California, Berkeley for a postdoctoral research career in 2020. And he is in his 3rd year of postdoctoral research in Dr. Gronert's lab at Herbert Wertheim School of Optometry and Vision Science. During his postdoctoral career, he has worked on understanding the role and regulation of neuroprotective lipid mediators in the retina and optic nerve under neurodegenerative conditions. Shubham's scientific vision is to discover novel targets and find putative treatment options for ophthalmic, really giving me a mouthful here for all of us, diseases by utilizing the potential of cutting-edge technologies in molecular and computational biology. So please take it away.
Thanks, Daniel, for finding introduction. So today I'm going to talk about a neurodegenerative disease, which is glaucoma, and some novel findings that we have from our research group. So starting from the very basic thing, that what is vision? To understand vision from a very layman's perspective is nothing but an energy conversion. It's a conversion of light energy to electrical energy. What you are seeing here is a cross-section That means if I'm standing like this, you're seeing my eye like this and the brain in this direction. And when the light comes in this direction towards me, light enters the eye. The eye is the center where the light energy gets converted to the electrical energy. And these electrical impulses are then transferred to the brain via something called as optic nerve, which connects the eye to the brain. The major thing is that our eye does, it converts the light energy to the electrical energy, as I have mentioned. So later on, we are going to study eye anatomy. Why this is important? Because, so again, same, we are seeing the cross section of the eye towards, suppose if I'm standing like this, you are seeing the cross section of the eye in this direction, and the light hits me in perpendicular direction like this. And what we are seeing is a cross section of eye, and this red area marked here is called retina. Why this is important? Because retina is the segment in the eye which converts the light energy to the electrical energy. Why this is important? Because retina is made up of multiple different cells. That means retina consists different cell types. Like our body is made up of millions of cells, retina also contains some different kinds of cells. And these cells are always arranged in a certain manner. This needs to be arranged in a certain manner to function properly. Now, to understand this more accurately, let's take this example of a Lego. So this Spider-Man or neighborhood Spider-Man can only function when all the components of this Spider-Man is assembled correctly. This same phenomena works with the retina as well. Retina has to be assembled in a certain manner with certain cell types to function correctly. That's how it converts the light energy to the electrical energy. Now coming to the disease, which is glaucoma. Glaucoma is the second leading cause of blindness worldwide, and it is currently affecting 80 million people, and it is projected to affect around 111 million people by 2040. On the left side is an example of how a person sees during glaucoma. What happens is our peripheral vision, if we are seeing something, so whatever we are seeing in the center is always remains focused, but we start to lose our peripheral vision. That's what we are seeing here. The dog is in focus, but around the dog, everything becomes blurry. That's what starts to happen in the glaucoma. That's how a person sees in the glaucoma. There are tests available that your provider can do to test whether you have glaucoma or not. There are certain therapies available in form of either eye drops or either surgeries, but the treatments won't undo the damage that glaucoma has already done to the eyes, and also most of the times these therapies doesn't work efficiently to provide or to rescue the vision that you have so that you do not lose it further. So that's here where so let's first also understand what actually happens in glaucoma. So our eyes have certain anatomy, certain shape. It's a round eyeball, right? So it has a round shape because it has a fluid inside it. That fluid, it constantly being generated inside the eye, and it also exits the eye. That's a natural phenomenon that happens in a normal healthy eye. The fluid is secreted, it gets out of the eye, and eye has a normal pressure. What happens in glaucoma is, for some reason, imagine the outflow of the fluid is hampered or is not happening, but the fluid is being constantly being secreted. What happens is there is more accumulation of fluid inside the eye, which it starts building the pressure inside the eye, and hence it starts killing the retina. Retina starts disintegrating, it kills the retinal cells, and we lose the ability of our eye to convert the light energy into electrical energy. So here our research comes. Few years ago, around in 2017, our research group, when I was not in the lab, they took a chemical compound. They treated the rats with the glaucoma with that chemical compound, and they observed that the rats are no longer having glaucoma. Glaucoma, the progression of the glaucoma stopped. But the question is, what is that magical chemical compound did they use? That compound is, we know, in our daily to daily lives, we eat a lot of fats, right? Uh, Whenever we go in grocery shopping, whenever we buy something, there is a label on the back that says fats. It says saturated fats, monosaturated fats, and polyunsaturated fats. So the polyunsaturated fatty acids are actually good fats. These fatty acids are present in fish, walnuts, flax seeds, and other compounds, other foods, and the oils derived from them. When we consume these food rich in omega-3, which is a polyunsaturated fatty acid, our body has ability to produce that chemical. That chemical has a name. This is called as lipoxins. And interestingly, what they found is whenever there is a disease condition in the body, our body loses this ability to produce lipoxin. No matter how much omega-3 rich food we eat, our body won't be able to produce lipoxin, which in turn gives us the protection in the disease. So now here comes the scientific problem, that how do the lipoxins work? Because if we want the lipoxin to come from research to a provider giving, prescribing this as a drug to the patients, we need to know how these lipoxins are actually working. Here I come. And so to start with, what do I do in the beginning? What do I do in the beginning is we use mice in our experiments. I treated the mice with lipoxins, and then I asked a very basic question. I took the retina from the mice, the retina that we originally discussed as a model of assembled Spider-Man. So I took retina from the mice and I asked very simple question is, what component of retina is actually responding to the lipoxin treatment? And to understand this, I used a cutting edge technique, and then my results showed is only one cell type in the retina was responding to the treatment of lipoxins, which is named as microglia cells in the retina. Going forward, I would address that what cutting edge technique did I use and what are microglia cells. Now coming to the technique, this technique is called as single cell genomics. What happens is, so our human body is made up of millions of cells, and different cells, all the cells carry same genomic information, but they carry different information at different levels. Like they carry different proteins, they carry different lipid inside the cells. So to understand this, imagine by this model is like each house in the society is an individual cell. What this technique allows me to do is to hear inside each of the house is what conversations are happening, so that I can understand that, okay, in some house people are fighting, in some house people are angry, in some house people are diseased. So it can allow me to understand informations from every single house, that is from every single cell. That allows us to understand the disease progression at a very deeper level. Now coming to what is microglia. As we know that we have immune cells in our body, in our bloodstream. So similarly, microglia are immune cells of brain and the eye. They are present in the retina, in the optic nerve, and in the brain. In the healthy individual, The cartoonic representation of microglial cells are something like this, and they are happy. But what happens to these cells in the disease condition is they change their morphology, they change their shape. Now, from that morphology or shape, they become more circular, they become angry. In the healthy states, their job is to eat the dead cells in the body, to keep everything healthy. But in disease conditions, they start killing other cells. So their role is not changed. What happened in my research is, going forward, what I did is I took lipoxins, I treated the mice with glaucoma, and then what I observed in my results is, in disease conditions like glaucoma, microglia was angry and it was killing other cells. But upon lipoxin treatment, it became healthy in the retina and the optic nerve. And it starts doing its normal thing, which is eating the dead cells. That's how we figured out that lipoxins are giving protection in glaucoma by affecting the microglia cells in the eye, especially in the retina and the optic nerve. Why this is important? There are many brain diseases like Alzheimer's, Parkinson's, multiple sclerosis, where microglia cells are involved. Similarly, we can extrapolate this finding of lipoxins, and we can start testing this thing in the other brain diseases as well, whether this can do a similar thing in the other diseases. That's why we are going towards the other diseases, looking what lipoxins can do. So, in summary, as I have mentioned, lipoxins are derived from polyunsaturated fatty acids. So keep eating your fish, walnuts, that's good for you. And then lipoxins provide protection against glaucoma, and lipoxins affect microglial cells. To acknowledge and some of my talk, I would like to first acknowledge the mice that I use in my studies. And then my lab members in center is Dr. Gronert, is my supervisor and project leader. And here I am. And we have tons of undergraduate students who work with us. We have graduate students who work with us. And we have a generous funding from National Eye Institute, Bright Focus Foundation, Glaucoma Research Foundation. And last, I would like to thank organizers, Open Science Global, and Wertheim. I would leave this slide on for questions if you have this slide. This QR code will lead you to the NIH glaucoma page, where you can have basic information about glaucoma treatment options and what current research is being done and being funded by NIH. Thank you.
Questions?
I have some, but do you have any? So that was fascinating. I have glaucoma. I just got the diagnosis, and I just happened to be walking by and I was like, oh, glaucoma, I have to sit down. And so this was really helpful. I thought I heard you say that in a healthy body, eating the lipoxins through diet, fish, walnuts, other sources, is efficacious. It does help the eye, but when you get the disease, it stops being efficacious. The body can't take in. Did I hear that correctly?
Yeah, correct. So in that situation, so that's totally correct. Our body, when we eat the fish and food rich in omega-3, our body makes this lipoxin. It is not only connected to glaucoma, it is connected to many other things in the body that I did not talk about, but it also provides protection, or it also provides a normal what do I say it? It's like our cells do normal daily activities, right? In our eyes. So what happens is lipoxin is constantly being produced in the eyes. So, and that helps our eyes to do its daily to daily activities. But in glaucoma, our eyes cannot produce lipoxin, and then our cells lose ability to do its daily abilities. Apart from dying from the pressure inside the eye, they start to feel these side effects as well, because now they don't have like toxins to protect them. That's why when the researchers in our group did like applied lipoxin externally, like your body, when you eat the food, your body metabolizes the food and then breaks it down to lipoxin. That's the process. But as a drug, you can directly take lipoxin. And then what happened is the progression of glaucoma stopped, like it should happen if lipoxins were originally there. If I answer it.
I think so. I think so. But I guess the follow-up question is, and maybe this will show that I didn't understand what you said. So the application of lipoxin it's derived from some source and you're administering it to the eye directly, not in a dietary manner, and it acts at a chemical level immediately on the eye?
Yeah,
Is that what's happening?
In our experiments, we did like we used eye drops
Uh-huh.
to directly provide the lipoxins, and we also administered to the veins.
Oh, interesting. Intravenously. Uh-huh. So I'll keep asking questions, but if you guys have more with it.
I have a question. So at the very beginning, you said that a lot of the treatments right now, either is like through eye drops or like surgery, it's not very effective and often, leaves the symptoms unresolved. So would you suspect that like if you apply lipoxin, clinically to patients, how that will be different from the other like eye drops or, surgery?
So the current method available is to reduce the eye pressure. That still does not change why the pressure is being built, right? But what lipoxin does is, even though there is a pressure, it stops, it provides a protection to the cells. It's like provides a shield to the cells.
I see.
Because what happens in a general condition is like this eye lowering drops, you have to apply thrice a day sometimes. Sometimes you have to apply twice a day for weeks. So people miss those times. So it's not very efficient in that manner. But lipoxin provides a shield, even though the pressure is being built in the eye, but your cells are protected. The retina is not done.
Would you mind talking a little bit more about, so the lipoxins help these cells be able to identify the dead cells and get rid of those. Would you mind connecting that a little bit more to, you mentioned how glaucoma also comes from kind of that buildup of pressure because the fluid can't get out. So how does kind of being able to properly destroy dead cells connect to that buildup of pressure
Dead cells are cleared by microglia. Microglia are cells inside the retina, and the lipoxin helps the microglia to clear the dead cells. So lipoxins are not directly clearing the dead cells. Lipoxins are regulating. Lipoxin is a chemical molecule, and microglia is a cell type. So that chemical molecule is talking to the cell and making it do things. That's why
it turns the switch on.
Yeah.
Okay, got it.
and so that's able to like clear debris or something so that the fluid can leave?
No, fluid is still there. We don't know right now that whether fluid is able to leave or not, or research is not at that level. But we can say that the pressure is still there. But what happens is, so when there are cells dying because of the pressure, because of the death of the cells, is the environment becomes toxic.
Okay.
Right? So the toxicity leads to more death. It becomes like a add-on thing to the pressure that you already have. And the things add up, and then the dying process, the rate increases. So lipoxin talk to microglia. Microglia starts clearing the debris, the dead cells. So the process of dying is halted somewhere in between.
Thank you.
So with what you're doing now, what are the next steps to bring this to people like me who are like, yeah, bring it on. Help me cure my glaucoma.
Yeah, so now we know that a cell is a target. We now know that microglia is a cell in the retina, which is a target for liposome. Now we need to know in detail that, like, inside a cell, there are so many things happening at different levels. So we need to understand at the more deeper level what, like, we call it cellular pathways in scientific terms. So we need to know what are things being changed, or should there be any side effect of this if it is coming in the human trial. So we need to know, we need to clear our like background very thoroughly, and we need to search for, like, if there could be any potential pitfalls, or like how can we increase the efficiency, what doses are required. Like right now it's in mice, we are optimizing the dose as well. So those things we have to first figure out, then we can go to the clinical trials.
Do you have to bring the mice research through your institutional animal review board, or you do? Are you involved in writing the protocol for that, or?
So when I joined the lab, the protocol for this was already written.
Okay.
So this protocol was already approved by institution. So for this project, it is approved with next year.
Got it.
Awesome. Let's do one more round of applause.
Yeah.
Switch over to our next speaker.
While Haley sets up, I'm going to introduce our next speaker, Haley Frey. Haley grew up in New Jersey and then moved to Tennessee for college to study neuroscience at Vanderbilt University. She picked up some research in object recognition and decided that she really enjoyed learning how the visual system makes sense of the world. And Haley completed a senior thesis in object recognition and then moved to Michigan to work as a research assistant at Michigan State University. And there she learned how to analyze fMRI data and was introduced to the topic of change blindness and how consciousness plays a role in vision. And finally, Haley joined the Vision Science Department at UC Berkeley in August, where she has continued research in visual perception. Outside of research, Haley likes to craft, play volleyball, and tend to her plants. So without further ado,
I'm working on it.
yeah, take your time.
There we go. I'm a vision scientist, not a technology expert. Okay, thank you all for coming to my talk today. I'm going to talk about visual perception, and my talk is titled "To See or Not to See? How does the brain decide what we see?" So I'm going to start with a little bit about me, which is just read in my bio because I forgot that was happening. But what makes me qualified to talk about visual perception? So I got my start at Vanderbilt studying object recognition and was introduced to how complex the visual system is and how much work goes into being able to see my presentation and just think about what I'm saying and think about what you're seeing on the board. After graduating, I moved to Michigan State University. There we go. The internet has been a little laggy. And here I got into thinking about what information do we see and what information do we not see? Because lots of things are going on in the environment, but we can't be consciously aware of everything all the time. And I actually just published a paper on the work that I did there. And now I'm at Cal. I just started my first year in the vision science department. I work with Dave Whitney. He's over in the psychology department, so I'm kind of 1 foot in both worlds there. So for today's story, how does the brain decide what we see? So we're going to go into 3 topics. We're going to learn about perception, attention, and serial dependence, and how these can all help answer the question of how does the brain decide what we see. So I'm going to begin with a demo that I think is going to serve as a scaffold for talking about all these topics today. So all of them are going to relate back to this. So while you're watching this image, I'm going to present it on the screen. Some parts of it may change. And so over the next 20 seconds, keep an eye out for any changes you notice, and then we'll talk about them after. It's lagging a little. There we go. Okay. Raise your hand if you notice at least one thing change. Okay. Keep your hand raised if you notice at least 2 things. Keep your hand raised if you notice 3 things. Anybody notice 4 things? No? Okay. So there are actually 4 changes that happened in that, and we'll go ahead and break it down. So first what happened is the rocket ship in the top changed from tilting one direction to the other. The shoes of the little boy changed from red to black, and then there was no radio in the corner, but there was a radio in the corner at the end. But also the color of the car changed from orange at the beginning to yellow at the end. Did anyone notice the color of the car change? You've seen this video before. So how is it possible that while you're looking at this image and you're paying attention and you're looking around, that you fail to see these changes happen right in front of you? Well, you're about to find out. Hopefully. There we go. I hope it's not too laggy for the people on Zoom. Okay, so what you just experienced is something called change blindness. So this is when observers fail to notice large changes that happen right in front of them. So there's 2 types of change blindness. Classic change blindness, like the rocket, where the change happens quickly and you don't notice it, maybe because you weren't paying attention or because you hadn't looked at that part of the image before it changed or after it changed. Then there's also slow change blindness, like the car example. So the change happens so slowly that even if you're paying attention to it, it can be hard to tell those differences.
It's changing on Zoom.
It is on Zoom?
Yeah, it's changing on Zoom as well.
Oh, yeah, okay. I'm just standing here after. Oh, here we go. Okay, so to answer our question of how does the brain decide what we see, as I mentioned, we're going to talk about perception, attention, and serial dependence, and we're going to relate them back to this example. So perception is the brain's ability to make sense of information from the senses. So when we're talking about vision, we're talking about the brain's ability to make sense of information coming in through the eyes. Now, I don't mean to scare you with this figure. This part right here is the brain. This is a similar image to what our earlier speaker showed, but from a different angle. So this is as if you're looking at my head like that, like someone chopped it like that. But I just wanted to show here that information comes in through the eyes, and then there's a lot of connections that need to be made before the visual information is processed, and before we even know what we're seeing, and before we can even do things and act on what we're seeing. So how does perception relate to change blindness? Well, as you can see, there's a lot going on in this image, and something has to convert the signals of the orange of the car and the placement of the rocket ship and how everything relates in the scene into something that's meaningful. Something has to let you know, oh, that's a car. Those are people and things like that. And this is what is going on in the brain. Okay, we move on to our second demo. This one may be a little easier, maybe a little harder, we'll see. So there's going to be two images that continuously flip back and forth, and there's going to be a gray screen in between, but it'll just kind of loop on repeat. So there's only one change, and I want you to clap when you notice what's changing. So it may take a little while, but when you notice what's there, you can go ahead and clap. Is everyone ready? No one? Are you guys even trying?
Trying.
All right, let's pause for a second. So clearly this is pretty hard, right? I'm going to give you a trick that might make it a little easier. Pay attention to this area right here. All right, I'm going to show it again. Remember to clap when you see it. Yeah, okay. So by now you know it's this very obvious jet engine changing. It makes you feel stupid that you hadn't seen it before. So this gets to our next point of attention. So attention is the brain's ability to prioritize relevant information. So this is how you know what's important to you at any minute and helps you filter out some of the background information. So for example, right now, you're paying attention to my talk, hopefully, and not paying attention to the feeling of your toes in your shoe. And now you are, maybe because I just drew your attention to that, but because in order to be able to pay attention to my talk and to take in this information and think about it, you need to not be paying attention to the feeling of your toes wiggling around. And so in the previous example, attention helps you to identify where the change is happening. And so if you were to eventually watch that video for a while, you would move your attention around to different spots and see it. But I didn't want to be here all night, so I figured I would draw your attention there. So within attention, there are two types. The first is top-down attention. So this is like when you're doing a Where's Waldo. You know that he's got a red and white striped hat, red and white striped shirt. So when you're looking around, you're looking for that information. You know what's important to you and not moving. In contrast, we have bottom-up attention. This is when you're walking home from the grocery store and you're focused on getting home because you're really hungry and you've got a bunch of food on you. And a fire truck goes by and it's really red, it's really loud, and it grabs your attention, whether you wanted it to or not. So in this case, the stimulus itself is saying, hey, I'm really important, you need to pay attention to me, and it's going to capture your attention. And so in perception, visual system is constantly having to balance between top-down attention and bottom-up attention. Okay, and so how does attention relate to change blindness? Well, we kind of answered this already, but if you were not paying attention to the area of the screen where the rocket changed, you may have missed that happened, which this still might not answer the color of the car changing, because even if you were paying attention to the car, that color changed. Given that it's so slow, it's pretty hard to detect. All right, that brings us to demo number 3. So this video has some text that goes along with it, so I'm just going to read it out loud. It should be pretty straightforward. All right. Okay, so these are two identical twins, and they're the same age. How old do you think they are? Just shout out a number. How old would you say these people are?
4.
Like 4? All right.
Teens?
Teens, okay.
Young teens?
10, 11. Okay, well, keep in mind, however old you think they are, how old you think they are right now. Okay. Okay, now one's going to go behind a curtain for a little bit. Maybe she's taking that. All right, so I want you to keep thinking about how old this person is while you're looking at them. Keep looking. Keep looking. Maybe they're aging a little bit. Yeah. All right. Does the person look a little bit older now? How much older than before would you say that they are?
10 years.
10 years? Do you think they look? Anyone think they look 5 years older? No. 5 or 6 years older, 10 years older, maybe 20 years older, much older. Okay, that's not the answer I want here, but you guys are trying your best. It's okay. Okay, so this is where the person was when they first started aging. Now, is that younger or older than you remember them being?
Younger.
Do you think that they've aged more now that you see the comparison? Yeah, right. So this is an illusion that I'll talk about in a second, but yeah. Okay, so now I'll explain the solution. Okay, so this is called serial dependence. Serial dependence is the brain's use of recent information to influence our current perception. So for example, this is a picture that I took of a river and some trees. And from moment to moment, even like days to days, sometimes weeks to weeks, this scene is going to look largely the same, at least on the big scale. There's a big chunk of green over here with some trees, green in the middle, green on the side. And so there's a lot of regularities in the world and things that are stable and not changing from moment to moment. And so the brain will capitalize on this because it takes a lot of energy to convert all the visual information that we're seeing into something meaningful at any time. So instead of each milliseconds, taking a snapshot of information, processing all of it in really high detail, instead, the brain will be like, okay, I know there's green, I don't need to give as much processing to that anymore unless something really changes. So we can kind of accumulate information across a period of time to generate the representation instead of doing everything over. So in the previous demo with the twins, while you're watching the face age, it may appear that they're aging at a slower rate than they actually are because you have information from them being younger as they age. Meanwhile, if you were to just see the final image, aged face, you may rate them as older than without the gradual aging beforehand. So how does serial dependence relate to change blindness? Well, the video that I showed you is about 20 seconds long. And maybe your brain's like, well, cars don't really change color. And from moment to moment, between like the first 2 frames or the second 2 frames, not a lot's changing. So perhaps instead of refreshing the color of that car every moment, maybe the brain is taking an average of that information and just having a single representation, and that's why we don't notice the change. So all the explanations I propose today are, of course, up in the air. This is what people are like myself are researching. So yeah, change blindness is something that I study in my research using these stimuli. And so I've done some work trying to figure out what people represent at the beginning of the video, what people represent at the end, whether they have a representation of the beginning color or the ending color, or maybe both, and they just don't compare them. But right now, my interest is in how do we perceive the color of the car at any moment? So while the color of the car is changing, what does it look like? So maybe serial dependence is acting and we have some sort of intermediate representation. I think that would be really interesting because it doesn't match what's actually on the screen. And it's interesting to think that, we're reconstructing the world. So what you perceive might not be what's out in the world. So just some take-home messages for my presentation today. To answer the question, how does the brain decide what we see? Well, we talked about 3 things. First, the brain uses perception to reconstruct the world based on information from the eyes. So it's not what you see is what you get. You have to work hard to generate that. Second, the brain uses attention to help determine what information is relevant at any time, because it wouldn't be helpful to be paying attention to everything all the time, or we would never be able to get anything done. And lastly, the brain uses serial dependence to represent the largely unchanging world efficiently. And this is really great most of the time because we can save some brain power. But if you're ever in a situation where you need to identify slow changes, it may hold you back. I just want to go ahead and acknowledge the Braskamp Lab and the Whitney Lab. So this is my past research group and current research group, and in particular, Jan, who's in the white shirt there. He and I gave a slightly similar version of this talk at the Michigan State University Museum last year. You can find out more information about their research at those links. And then thank you all for giving me your attention and paying attention to my talk. You can email me with questions, comments, and concerns, or check out my website with QR code here or the link. Happy to take any questions.
Very cool talk. I have a question about, so the first video that you saw, that you showed is like a colorful, like change of like scenery.
Yes.
Would it be like easier or harder to detect if it's like black and white or grayscale?
That's a good question. Something that we've talked about with my colleagues in research is sometimes people are sensitive to other components of light and color. So if something were to get brighter, that's one way that visual information can change, or in color. So if it was black and white, you wouldn't have any color changing information, but you may still be sensitive to the luminance. But even if it's happening at such a small scale or so slowly, I think it would be difficult to see, to notice. Yeah.
Kind of a similar question as well. So the video that you had at the beginning, it was kind of like an amalgamation of a lot of different things. Like I saw like the boy had like a robin in his bag, and like the mom I think was like holding a house as a purse, and so like those are kind of, you know, a little bit more strange or abstract, non-everyday things. And so is there like a correlation with being able to perceive more changes with like if they were normal, if it was just like a regular purse that was changing color, or like not a rocket ship that was in the sky? Or
That's a good question. That image in particular is quite busy. As I mentioned, I use images like that in my research. Some of them are a lot more sparse, and it's really just the large color changing region, and people still don't notice it. But there has been some research done, at least for those quick changes, that shows that changes that are like more relevant or semantically meaningful to the scene, people can notice those more easily than ones that are not. So for example, if the identity of an object changes. So if it was a kitchen scene and the blender on the counter went from a blender to a Keurig, maybe that would be more easily noticed than something to change color or to change size.
So how do the like perception would change for a colorblind person?
Yes, that's a good question. In my research, I filter out people who are colorblind, so I don't have any data on that. But I think that would probably go back to the luminance point that I made earlier, that even if you're not sensitive to color, you may pick up on luminance differences. And if those happen quicker or over a longer period of time, you may be able to notice those as a color normal person would be able to pick up on some more extreme color changes. But yeah, as for a video like this, I don't know.
we do have a question from Zoom. What was the actual age difference between the twins?
Oh, I didn't get to that part of the video. 10 plus ages, I think. Yeah, so the end of this video shows with an age counter. We can actually go ahead.
Did we see the entire? Can we see this entire segment? Yeah.
This whole video, yeah?
Yeah, I didn't catch the significance of the serial dependency.
Sure. Okay. Goodness. Okay. So we start with identical twins who are the same age. And the idea is that we're covering one up so that her face stays the same, and we're going to age the other one. And so while you're looking at this face, similar to the color change, the slow color change video that I showed earlier, this change is happening slowly while you're looking at it. So you may not even notice that she's changing very much, if at all, until the end. And so in the research paper that they wrote on this, so this is the lab that I'm in right now, they showed that if you were to just show people the like ending phase, they would rate it as much older, like maybe 10 years older, but having been preceded by the slowly aging video, so starting young and then eventually ending at that same age, because you have all of that visual information of a younger face, kind of pulls that perception and that average to make the person look younger. So here is the age counter alongside the same video. Does that make sense?
So if it was just one face, we would be able to see it more accurately. change.
Yeah, so if I just showed you the two ending phases, you'd be able to, you'd probably rate the one on the left as being much older than the one on the right, or even if it wasn't a comparison and just looking at that face, maybe you could say they look, I don't know, 20 years, something like that. But then having watched a bunch of visual information of a younger face, your perception of the face is that it appears younger to you, even though it's the same stimulus. Yes.
Is there a wide disparity in people's ability to recognize the perception? Like some people can see typos quickly, and other people are more bigger picture. Do you find that some people saw all four of those changes, and a lot of people didn't see any distribution?
I'm sure there are individual differences. Off the top of my head, I don't know. In my research, there did tend to be people who either saw, at least for the quicker changes, either all of them or maybe just one or two. I don't know if that correlates with their strategy for going about it, or if there's different expertise levels of visual processing. Certainly if some of the information is more relevant to you, as we talked about how the semantic content can play a role. If you are a soccer mom driving a minivan full of kids where there are rockets in the sky, maybe you're gonna be more likely to pick up on things like that. So that bottom-up attention of things that are relevant to you may grab your attention, and you know, house for a purse might not, because it's not really relevant for your day to day.
Yeah. So, yeah, I remember my question now. For the first and the third demo, when you're like, you know, detecting change, or no, the second, sorry.
Yes.
The jet engine and also the like changing scenery, we were, you know, kind of directing our attention like based on what we want to do.
Mhm.
But if you were to say like you have to fix it at the center the whole time, would you expect people to be more likely or less likely to detect changes?
I think this is a good opportunity to discuss the distinction between like eye movements and attention. So most of the time you're paying attention to where, sometimes you're looking one way and you're paying attention to people talking over there. So when you're given free range or the ability to preview a video like this one, your attention is probably where your eyes are going. But if you're asked to fixate on the center, then you're kind of like tending out of the corner of your eye. You're still trying to like, you know, sample different areas to find that. In this case, the jet engine is pretty close to the center, so maybe you would see it more quickly having your eyes in the center. The nice thing about being able to move your eyes around is that your highest acuity, your best ability to see is right where you're looking. And so if you're asked to look right at the center of the image, now things in your periphery are not going to be represented in as much detail. So that may make it harder. That's good.
Alrighty then, let's give one more round of applause.
up our finishing slides. But thank you again for coming out to this month's Popping the Science Bubble. And again, thank you for coming out in the rain. I know that was certainly not an ideal time to be coming out, but we very much appreciate it. Our next presentation is going to be on March 19th, Tuesday, at 5:30. Once again, right here. Just looked up and I can show you. There we go. If you want to join our mailing list, you can use our QR code right here. We have Twitter, we have Facebook, a YouTube channel, which these two fantastic speakers are going to be on there very shortly. And as I said, next Tuesday, not next Tuesday, March 19th at 5:30, which our next speakers are about.
Okay.
Or if you don't have it handy,
No.
totally fine.
One is about dark matter, and the other one I think is like conscious, like building a mechanical mind from the electrical engineering department.
All right, very cool. So stay tuned for next.
To the recording.
Thank you.
Thank you, guys.